Okay, so I just came across something wild. A new model called la-proteina that can generate fully atomistic protein structures with their sequences.
Wait, seriously. How does it manage to do both the structure and the amino acid sequence at the same time?
Right? What's fascinating is that it uses a partially latent representation to connect coarse backbone structures with detailed records on each residue.
That makes sense. So, it avoids the typical headaches associated with representing side chains, which can shift dramatically during generation.
Exactly. It's like they put together a straightforward blueprint while still capturing all those tiny details.
This really shifts how we approach protein design. But doesn't it assume we already understand the biological implications?
For sure, but think about it. By focusing on the backbone initially, they can simplify the more complex aspects.
True, though. The protein's functionality heavily depends on those side chains, so how does it ensure accuracy there?
Good point. They tested la-proteina against benchmarks for co-designability and structural validity, and it performed really well.
That's impressive, but I'd love to know how it stack up against earlier models. Any surprises there?
Yes, it even outperformed previous models in scaffolding performance, which is crucial for design tasks.
Wow, so it can design proteins with 800 residues. Most models just fail beyond that.
Right, that's a game changer. Imagine the novel proteins we could create, but can it really maintain robustness at that scale?
It's worth contemplating. Scalability is one thing, but sustaining accuracy is another. Any limits in their results?
Yeah, it may still face issues in certain edge cases, but the groundwork they laid feels solid.
And it's cool that they encourage community input by providing an exhaustive setup guide for using the model.
For sure, whether using Mamba or Conda, they've made it accessible, which is critical for broader exploration.
Totally, but what about the actual data they use for training? Does it lead to any biases?
Interesting question. They specifically trained on subsets of the AFDB, not just any protein database.
So that's a targeted approach. Still, the implications of relying on just one database could be significant.
Right, and while it's impressive, the diversity of protein structures in nature is vast. Are they limiting potential by this choice?
That's my concern too. But the model's architecture is designed for optimization, which could help balance that.
Agreed, the use of transformer architectures could unlock new capabilities as they refine the model.
Speaking of refinement, how do they handle the evaluation of generated proteins? What's their method?
They use protein MPNN for computing designability, which sounds robust for validation.
Smart, so it's built to evaluate how functional these generated proteins actually are.
Exactly, and they also provide options to run several scaffolding tasks, which broadens its utility.
This whole discussion just shows how intertwined structure and function are.
Absolutely, and it also raises the potential for ethical concerns. We're generating life forms after all.
Interesting point. It opens up the discussion about how these models could impact biotechnology.
Right, there's an exciting line between innovation and responsibility, even in tech like this.
I can see this leading into major breakthroughs, but we need to tread thoughtfully in how we apply it.
For sure, so if we wrap all this up, la-proteina signifies both promise and a responsibility to proceed cautiously.
Definitely. What's fascinating is the potential for new discoveries, but the conversation shouldn't just stop here.
Exactly, readers and researchers should ponder how we balance discovery with ethical consideration.
There's a world of possibilities ahead. I can't wait to see where this leads.
Me too, and to think this is just the beginning, imagine the innovations we'll uncover next.